And happy Thursday morning, everybody. So what I want to tell you about, I want to continue on things that I was talking about a couple of days ago, looking at biofluid dynamics at the microscale with regularized Stokes-Lit formulation. And today I want to talk about two projects that that we've been recently working on at Tulane: confined helical swimmers and coupled oscillators. So let me tell you first on this confined helical swimmers. Here are my three collaborators at Tulane. And John Legrone is a postdoc. Ricardo is my colleague in the math department. And Kyriakos Papadopoulos is an experimentalist chemical engineer. And so briefly, when one is looking at how bacteria swim, one thing that is really intriguing is there are particular types of bacteria that have more than one flagellum. And you can see here, these are E. coli. And possibly you could see that during that these many helical flagellum at some point form a coherent bundle, which is called a flagellar bundle. And it's been known, at least for E. coli, that the way they swim straight for a while, and then something happens with one or more of their flagella, and it turns in a clockwise direction versus counterclockwise or some such thing. And the bundle actually flies apart. You can see right here. This isn't such a great example, but that causes the bacterium to change its direction. When it has a coherent bundle, it swims straight. Then when there are one of these tumbling events, one of the flagella actually, one or more, goes in a different direction and causes turning. And it's been shown that this process allows chemotaxis to occur, so if I'm a bacterium and there's coffee over there, and I'm too small to sense a spatial gradient of coffee, so I can't just say, oh, it's coming from that direction. But I have receptors that could sense temporal gradients. I could tell if I'm doing better. If if I'm swimming and there are these coffee molecules that adhere to these receptors, and it tells me I want to keep going in that direction, so my probability of reorientation is diminished if my temporal gradient is getting higher. So that's how some types of bacteria steer themselves towards a chemical, up a chemical gradient. Okay, so now I'm going to tell you what we're interested in. And so here are experiments from Kyriakos' lab. I'm part of a research project that was funded by what's called the Gulf of Mexico Research Initiative. So if you remember, maybe, I don't know how many years ago it is, maybe 7 years ago, there was a very big oil spill in the Gulf of Mexico, a BP oil spill. So the company BP has given some funds towards research, a nice public relations kind of thing. So what I'm showing you here is an experiment in a lab in a microcapillary where Kyriakos looked at a packed bed of soil particles. This is actually cryolite sand. And what is in this abstract movie? What I could point out, you see some movement, like here, right? And here. So these are the bacteria that are swimming through this porous matrix, through these sand particles. Why are we interested in this? Why is BP interested in this? Because there are bacteria, both natural bacteria and perhaps engineered, really natural bacteria, but one could try to find species of bacteria that actually eat up a particular toxin and degrade it. And so this is called bioremediation, using bacteria to eat up the bad stuff. And so with a group of biologists at Tulane, they are actually studying how these bacteria that live on seagrass, they're called endophytes, I think. So how this symbiotic relationship between bacteria that live on the roots of seagrass, how well do they do eating up the oil in the Gulf? So this was there's so many questions scientifically around this process. So one thing that when we started on this project, we wanted to understand the mechanics of how bacteria move through porous media, and also how they could possibly reorient in these tiny pores between soil particles. And so many years ago, my collaborator Kyriakis in his lab, before microfluidic devices were easily manufactured, he had a method of taking micropipettes and somehow heating them up and pulling them so that they could be only 3 microns wide. And so his experiments were pretty interesting. And he showed that. So here's this great Academy Award-winning movie where it's a tiny micropipette. I think it's 3 microns in diameter, and you see these dots moving back and forth. But what's dramatic about this movie and somewhat surprising is that, unlike the E. coli that I showed you in the previous picture in the movie, here these basically don't change direction. So I was telling you that these flagellated bacteria can change direction by unbundling their flagellar bundle and then reorienting. So this is not so surprising when you think about it, but this was the first experimental evidence that geometric constriction doesn't allow these E. coli to tumble, to run and tumble. That they're going in one direction. If they want to go back that way, it's going to be complicated to flay out the flagellum and form a coherent bundle. And so most recently, this was 1995, these experiments, and there were experiments most recently in 2014 in Roman Stocker's group that showed that solid surfaces, not these tiny tubes, but that solid surfaces also prevented this kind of flagellar bundling that caused reorientation. So when we started this project, we thought that we would look at the same thing, that we would look at computational models of flagellar bundling and reorientation, and then do the similar simulations in micropores. So, here I'm just flashing this up again. I talked about the computational framework that we're using is we're going to model a flagellum or whatever structure we have as supporting a bunch of regularized forces, and we have fundamental solutions. So, getting back to the point at hand, the question was, before we start thinking about multiple bacterial flagella that are being actuated or turned by a rotary motor, we'll look at one. So what I'm going to talk about today just came out in PhysRev Fluids, and it was supposed to be a warm-up problem for our postdoc. It was supposed to take 2 months. It took 2 years. Well, he's been doing lots of other things. So this is the nature of mathematics and science, right? So we said, instead of first looking at how two helices that are like bacterial helices when they're driven by a rotary torque, how they interact and form a bundle, let's just look at one. Why not? And so the questions that we're going to ask is, if we have a helical object, a helical swimmer, how does flexibility play a role in its swimming? And then we'll also ask, what happens if we take the same object and put it, instead of in free space, put it in a pore, put it in a cylinder? Okay? 2 simple questions. And so the computational model, like I talked about the last time, is such that, I'm trying to remember what I talked about last time. No, okay, so in the previous, if any of you were, I think I recognize y'all. So the last time when we were looking at sperm flagellum, there we only modeled the flagellum as a centerline distribution of forces, okay, which were actuated by bending forces all along the body. Here, this is a little bit more refined, more detailed. Instead, what we're doing is we're representing the swimmer. We're discretizing the surface of this cylindrical helix. So here, now, there at every cross-section of this cylindrical structure, there are nodes around the circumference of the cross-section, and then there are a bunch of cross-sections here. And what we're going to do, because this is going to be elastic, we're going to take all of these nodes and we're going to connect them to neighboring nodes and nodes on other cross-sections by connections these links will be Hookean springs. So this structure is made up of a network of nodes connected by Hookean springs, and we will associate with each spring a resting length when the structure is in equilibrium, and we'll also associate stiffness constants to all of these springs. And together all of these springs and the topology of the connectivity with the stiffness constants, the macroscopic structure will inherit an overall bending rigidity, or EI for those of you who know that those symbols. So how are we going to? So here, unlike the sperm flagellum, the actuation, the bending moments are not going to be distributed along its length, because this is not a eukaryote. So we're going to mimic the way bacterial flagella are actuated just by placing a torque at its front. Now, for real bacteria, if you have one flagellum that's being rotated at its connection to the cell body in one direction, there has to be conservation of angular momentum. It has to be a torque-free system, so the cell body actually rotates the opposite direction. So again, I told you this was a warm-up problem, so we are ignoring the cell body, but nevertheless we want to conserve. We want this to be a force and torque-free system, so we put a virtual cell body in front where we apply a torque in the opposite direction. Okay, so we consider this is a very simple model of something. Okay, you can't always ignore cell bodies and so on and so on. I'm ignoring a lot of things. Now I should say that for bacterial swimming, the actual individual flagella of bacteria are basically rigid. So and the flexibility that comes into the bacterial swimmer is the hook there. So there's another appendage that connects the flagellum to the cell body, and that has some elasticity. And so that is something that's been studied in the last few years. People are really looking at that. But here, what I'm imagining now is that this helix, this construct that I have, is more or less representing the whole flagellar bundle. So it's a flexible helix that's being actuated. What happens? At time 0, let's say, we initialize all the node points of this flagellum at equilibrium. So what's going to start things moving? Then we apply this torque by putting in a rodlet at the front. So once that torque is applied, the body starts turning, especially near the front, and it will pull some of the springs out of their equilibrium length, causing forces to develop. And so we compute those forces due to the rodlet and due to the spring deformation, apply it to the fluid, and evolve our system like that. So here is a simulation of this swimmer where we're just showing the resulting velocity vectors in a plane. This is 3D. So you can see the wave passing towards the back and it's swimming forward. You can see there should be a dot that is showing the presence of this virtual cell body where we're applying the counter torque. And I won't be able to tell you right now whether this rotation is into the screen or out of the screen, but so no surprises. So what we're getting here is so what's input into this model and what is output? What is input is the strength of the torque, the strength of the rodlet, and the passive bending stiffness of, or all of the Hookean spring stiffnesses. Okay, so those are two parameters. It is in a fluid of a certain viscosity, and it is swimming in free space. Okay, so this is in R3. So here's, here are 7 different swimmers, and all that I've changed here, and now they're swimming down and not to the side. All that I've changed here is the rodlet magnitude, or the strength of the applied torque that we're putting at its nose. And so it is no surprise that the rodlet magnitude 7 is swimming much faster than the one of rodlet magnitude 1. So these are all non-dimensional numbers. So the thing to note is that if these were rigid helices, and this is Stokes flow, if there was no feedback on the shape of the swimmer from the forces, when I apply something 7 times, I apply a rodlet 7 times stronger, it should swim 7 times faster. Perfectly linear problem. So does that happen? And maybe some of you have a good eye. Not that you could measure 7 times faster, but if you have a good eye, it could be though in this problem. The interesting piece of this problem is that these helices, although they natively want to have a prescribed shape, if those springs were strong enough, they are flexible. So when you spin things, they might not have the same shape. So, yeah. Is the stiffness of the springs the same all along the tail? In this simulations I'm showing here, but there's no reason for that to be. I mean, we can change that. That would be an interesting project. So just here's so here are just some numbers, and there were 2 things. There were 2 knobs that we changed here: the rodlet strength, the spring stiffness. Okay, so here, what I want you to look at here, and so these Ks are the stiffnesses. So the 1200 is the most rigid one. The 75, the dark blue, that's the floppiest swimmer. And so here, notice the rodlet strength, we're applying a torque. And so given that torque, we could also measure the frequency, how many turns per second you would have. And so that does increase linearly, but it's not perfect because the swimmer is flexible. So the other thing that, let's look here. So here's on this axis is the rodlet strength and the distance traveled per revolution. So if it was a rigid filament, a rigid helical filament, then the distance per revolution should be just a constant here. And you see for the stiffest swimmer, this purple, it's almost a constant. But as it becomes floppier, the distance per revolution is not a constant, and here's the floppiest. Okay, so just to bring this back a little bit to experiments, there were experiments out of Paris from a few, well, it's not only a few years ago now, 2008, so 11 years ago, where they looked at a flexible fiber in a tank. So this was not natively helical. This was we wanted to be straight, and put it in a very viscous fluid and rotated that wire. And as the rotation speed got faster, you can see that different shapes evolved. Okay, so we have similar change in shape, which I didn't, in that movie, if you looked carefully, you could see, well, you could see that the wave number changes as you turn it faster. I seem to not have the slide. So I told you those are two parameters that even though we non-dimensionalized all of our lengths and our speeds, we still kept as separate in these simulations the applied torque and the stiffness. But now we know in elastohydrodynamics there should be a non-dimensional grouping that measures the relative importance of viscous forces to elastic forces. And in the study of microorganism motility, this is lovingly called the sperm number. And here, the sperm number to the fourth. Here we have the viscosity, the frequency of rotation, the length of the structure to the fourth power, some term that represents drag that looks at the aspect ratio, the length over the radius of the filament, and EI is the overall bending rigidity. And so even though we're only inputting stiffness constants of individual springs, we could then look at the macroscopic structure, bend it, and back out the macroscopic bending rigidity. So let me look at this number again. So the sperm number becomes bigger when the structure becomes more flexible, when the structure becomes longer, and the sperm number becomes smaller when you become more rigid, shorter, and less viscosity. So this number has not just been used to study microorganism motility when studying fibers and flows and so on. It's been called the elastoviscous number, the effective viscosity, and so on and so on. So now, yeah?
This number works for a large range of fluid viscosity, or is only for a short? I don't know, but because in the Stokes equations, you could basically rescale the equations. You choose a force and time scale, and the viscosity comes out of the equation. So I think there's a pretty large range. Now, for experiments at the microscale, I don't know how big of a range of viscosities you can have. I'm looking at Roberto because I just don't know. Factors of 10, factors of 100, yeah. So now I showed you that data where distance per revolution for all the different stiffnesses and all the different Routledge strengths. But now we're going to plot the solution, plot that distance per revolution, not for all those data points as a function of this non-dimensional grouping. And we're always very happy when we see all of our data collapsing onto a curve. And so here, as a function of the sperm number, meaning the ones out here are long and floppy, and the ones here are shorter and rigid. We see that we get fairly constant for small enough sperm number, and then we get this linear drop off. We have these points that you see here for all of these different stiffnesses. Here you only see 4 red points, but there are actually more red points, but they all overlap here, and so this is in the graphics. Why this has, in this range of sperm numbers, why we get this basically linear relationship, I would love for someone to explain that. We don't know.
Average.
Oh, it's the average distance per revolution. So what are we averaging over, right? So what happens is in each simulation, when you start the rotation, it takes some time for the springs to all get into their happy place. So right at the beginning, the distortion of the springs is at the beginning of the fiber, and it has to propagate elastically back to the end, and then you basically get a distance traveled per revolution. So this is, we wait until we reach that periodic temporal state, and then we average over all of the, average over, say, 200 frames per 1 revolution. Yeah?
If the sperm number goes to 0, maybe it's because the viscosity goes to 0.
Yeah, so in these simulations here, the way that we actually did, and there is a data point at 0 here, because what we did, we reformulated the problem where we specified the shape of the helix, computed the forces it would. So there's another formulation. Instead of specifying forces and computing velocities, we specify the velocity, compute the forces, and move it. So it's a related calculation. So I don't think, the way that I think about varying sperm number is the way that my experimental colleagues can do this. They can vary length, and they could vary applied torque.
But do you expect that the advance of the bacteria can go to 0 if the viscosity is not 0?
I'm not sure. The viscosity in the Stokes regime, so the viscosity is going to 0. Yeah, so in the Stokes regime, the viscosity helps the swimmer. So if it were a wire, so here's the thing, when it's 0, that means that you're infinitely rigid as well. So the real problem, if you are a completely rigid wire and you are going to turn no matter what, the viscosity is not going to hurt you. But what happens is that if you put a much bigger load by increasing the viscosity, and if you're somewhat flexible, you won't be able to maintain your helical shape even with that applied torque. So we could talk more about it. I've not thought about this non-dimensional grouping in the 0 viscosity limit. Right.
But it wouldn't, I mean, the analysis wouldn't work because it's Stokes flow. You need the viscosity. Yes.
Right.
There's no viscosity.
Yeah, that's true. That's true.
It's another space. You're in potential flow, in which other weird things happen.
Yeah.
So I don't think the argument would hold.
Yeah, that's true.
Right? Because if you are in a only viscous environment, that's what I think.
Mm-hmm. Okay. Let's see. Okay, so here's what I wanted to show you. So if I go back here, and so this is true. So John, our postdoc, gave me these graphs, and I'm like, hey, John, what happened with the blue curve? Where are these data points? What happened? Just fill it out. And he's like, yeah, no, the simulations broke. I'm like, what do you mean they broke? So here's what happened. So the blue dots are the stiffest, the blue dots were the stiffest, no, the most flexible fiber being actuated the fastest. And so here's a simulation that broke. So here, you see that this is classical buckling. So what happened is that it's a very flexible filament, and it was being actuated really quickly, and it could not maintain its shape. And so we don't have a whole bunch of production runs to analyze exactly when this buckling occurs, but recently, this is recent, right, 2017, there have been some experiments out of labs, I think, at MIT, where they're doing physical experiments of flexible helices, putting them in some viscous fluid and rotating and looking at this kind of buckling. And the work that's been done in these papers is looking to see at what sperm number does this occur, and also what are the geometric parameters of the helix. Does it occur for a larger radii and so on. Okay, so now let's take this, and I guess I lost. Yeah?
Do bacteria do buckles naturally?
So there are all different kinds of bacteria, and there has been very recent work on looking at the buckling, not of the helix, but of the hook, the elastic hook that connects the flagellum to the cell body. And there are bacteria, for instance, that only have one flagellum, and they have to reorient somehow, and they can't do this unbundle and bundle, so they have to flick their direction. And it's because of this elastic hook that I'm not including at all. So whether or not, but having said that, I think there are some experiments with actual bacterial flagella that they could propagate a kink along it. So these experiments were like 20, I could give you the reference for that. Okay, so now that was, what I just showed you was the warm-up to the warm-up problem. So now let's take this structure and put it, not in free space, put it in a cylinder. And, the regularized Stokes-Slit formulation or boundary integral formulations are wonderful in the sense that we're not discretizing all of space, like in a finite difference method, but we rely on fundamental solutions to Laplace's equation. And, there are closed-form solutions in 3 cases that I know of: free space, above an infinite plane, and outside a sphere. There are between plates, there are infinite series solutions and so on for other things, but not for a cylinder. So unfortunately for us, we had to discretize this cylinder and specify not only that not specify forces at points on the cylinder, but specify that we wanted the fluid velocity at the boundary to be 0. So within this framework, we could do that, but it's expensive. So what we did, and you're going to see in the next simulation, I'm going to show you a particular swimmer being put in cylinders of different radii. And so there's the race. And so we're applying the same torque to the swimmer of the same flexibility. And you see in this horse race, the one in the narrowest cylinder actually won. So, and what I don't like about this movie is that it shows you what we're really doing. We didn't discretize an infinitely long cylinder. We discretized a cylinder and kind of moved that discretization with the swimmer. Now, there have been not so recent computational models and analysis by Tom Power's group and Severio Spagnoli, where they looked at infinitely long helices in infinitely long tubes, and the helices were infinitely rigid. And they found similar behavior, that actually, unless you're in the most tightest tube, for a given torque, you actually go faster as the radius of the tube gets smaller. Okay, so that's kind of interesting. But one thing that here that is even more impressive, let's see, I have this data. Let me show you. Let me use words instead of those graphs. So here we are applying a torque and not an angular frequency. So the one on top got there faster for the same applied torque. But in fact, with that same applied torque, because it's meeting the resistance from the tube walls, it actually, its frequency is smaller than the others. So the actual distance per revolution is much more dramatic. So you understand what I'm saying? If you took this helix and you applied a torque in front, and you measured on your stopwatch how long it took for it to complete a revolution, the one on top takes considerably longer because you're only applying a torque and it's harder for it to complete the revolution. So we could measure the same sort of thing, and now we've added a third knob in our simulations. So besides torque strength, flexibility, we also added the radius of the confining tube. And I'm not going to tell you about all the magic. This paper just came out last week in PhysRev Fluids. We could also do some scaling with our sperm number where we add in a drag term that's based on the radius of the tube. And voila, all of these simulations collapse. Again, linear. So we don't understand. We still don't understand that. Okay, so in the time that I have left, I want to show you a couple of other curious things. And here's what we could do now, and I do love this movie. What we could do, our helix is not infinitely long. So in all of the previous analysis, since the helix was infinite, it could not be launched. It could only be launched parallel to the axis of the tube, right? Otherwise, it would intersect the wall. So now here, instead, we take this swimmer, and instead of putting it right down the center of the tube, we put it at an angle. And so the question is, what is going to happen? Is it going to say, oh, I better straighten out with the center, or is it going to hit its nose right into the wall? So we did these simulations. Another cool set of movies. So here's the swimmer in 5 different tubes, and we've suppressed the translation. And you see that they take nice excursions, and it's hard. You really can't see what's happening from this angle, but I'll give you another angle. And so here's what I'm going to do. I'm going to plot for you the trajectory of the counter-outlet. So let me show you. Well, not plot. I'm going to show you another movie that's going to make you dizzy. So what's happening is this. You see those 2 white dots that look like snake eyes? If it was coming straight at you, you would only see 1 dot, because that would be outlet, counter-outlet. So what I think that you possibly can see is that the trajectory in the smallest tube is starting to center itself. So in the most confined tube, the swimmer, well, now they all stop. Here they go again. In the most confined tube, the swimmer centers. In the largest tube, you can really see those 2 eyes here. And in fact, in the largest tube, it actually, we stop the movie here before it actually hits the wall. So here are the trajectories, and I should have put a star on where they start out. So in the smallest tube, we get centering behavior. In the biggest tube, it starts out here, and it starts going towards the wall. And there is some sweet spot in the middle where the swimmer goes forward and just rolls around nicely around the tube. I have no idea what. I would love to see the analysis of the, how does this depend upon sperm number, flagellar length, radius, and so on.
Have you tried with rigid clicks? Because that might be just a long transient, right?
It could be. So I'm trying to think. I don't think we did this askew we put in the rigid helix at the end, saying, "Oh yeah, why didn't we do that?" So it gets tricky here because this problem, the calculations here with the tube, are much more computationally intensive because we're discretizing the tube, and we have to solve very big linear systems. So, not an excuse. It's just that's a lazy excuse, but we haven't done it yet.
Mathematically, is it possible to think of this problem as an infinite line? Not, I mean, you said somebody tried with a half infinite, but is there?
With the tube, they've done that with it.
And the flat, like,
Right. So that's been done. An infinite tube with an infinite helix, but you can't ask the question about what happens if you start at an angle, because then you'd be touching the tube wall.
No, yes, but the infinite case would have some kind of a steady-state solution, which might be computable.
Yeah, I think so.
Unstable, perhaps.
Right, I think so. So that in those papers that they did some analysis there, because you could also make use of lots of symmetry. You could cut a slice through it, and it becomes really a one-dimensional problem.
Because one could think that the angle problem is like perturbation of the.
Yeah, that's true. I never thought about that, but it just seems like even at the outset that it's illegal to ask that, because if it's infinite, then you would. But maybe locally, I agree. No, nobody's done that. You should do it, or your students should do it. Okay, so in fact, there was a paper in 2015 by Schumann and Gaffney, where they did boundary integral calculations, not in a tube, but in between two planar walls. And we found this out way after we did the calculations, and they found similar behavior in the sense that if a finite helix was swimming between two walls, if the walls were far enough apart, it didn't much matter. But there was a spacing between the walls where if the walls were affecting the swimming trajectory, if the spacing was big enough, it would hit one wall or the other. But as they brought the walls in, it actually centered and swam straight. So this is a very provocative conclusion, in that could it really be true that if you have porous media and you want bacteria to get from one side of the porous media to the other, would it really be better to have tighter pores? Where if the pores are too big, these bacteria are going to slam their noses into the wall, but if the pore sizes were smaller, they could guide themselves towards the center. I have a hard time believing that, but
maybe it's more expensive to do that. I mean, there's some energy expended in this process.
oh, absolutely. And so for our flexible helices, we could calculate that, and I'm not showing you that. But you're exactly right, because like I told you before, for smaller pores, for a single applied torque, it takes much longer to complete a revolution. So yeah, there are all these other things. So I have a few more minutes, and what I want to end with is a really cool problem in abstracting the idea of synchronization. So I've been thinking about swimmers next to each other. So here's, what I showed you warm-up problems. We want to look at actual flexible helices, bundling, and so on and so on. And I should say, what we really wanted to do was to then look at simulations of bundling in free space, and then put the whole thing in a confined tube and see what happens, see what the steric forces would be that would prevent it. Okay, but we've got years and years ahead of us. So I've spent a lot of my career looking at synchronization of oscillators, sperm flagella, cilia, and trying to understand what are the hydrodynamic effects that lead towards synchronization. Here I'm showing you there's a very beautiful experiments out of Ray Goldstein's lab in Cambridge, where there are these algal cells with single flagella that beat in a power recovery stroke. And you can see here that, so usually in the typical algal cell, there are two flagella on the single cell that beat in some kind of synchronized manner. And the question has always been, what is causing the synchrony? Is it because they're attached to the same cell? Or is it hydrodynamic effects? And so he did these experiments where there were these particular cells that had only one flagellum, and then held these cells on pipettes and showed that they are, so here if you see synchrony, it's not because they're on the same cell, it's because they're sharing the fluid environment. And so I think if you look at the ones that are closest together, you see more of this beating in phase. Here you get more of an opposite phase. So over the last 15 or 20 years or so now, let me just skip this because I want to show you. And so we've had some simulations where we've tried to capture the internal mechanisms, and so here you see two cilia that were beating in opposite phase, and immediately they start synchronizing because of hydrodynamic interaction. So I'm not telling you what's in this model and so on. But more recently, there have been experiments out of the physics community that looked at interaction or synchronization of colloidal particles or colloidal oscillators. And so I just want to show you some cool results that they had and some observations that we've made. So here's and these are realized experimentally and not just theoretically. So here's what happens. They take this colloid, this red dot, and put it in between two traps. I think they're magnetic or optical, I don't remember. I'm a mathematician. So here, there is then a force, let's say it's a magnetic force, that's taking this object and pulling it towards this trap. And the force is, think of it like a spring force. It's a quadratic potential. But then they can engineer this system in a way that they can measure the geometry. They could measure how close this colloid gets to this trap. And when it reaches a threshold distance, they turn off this magnet and turn this one on. And so here, there's a force like a spring force pulling it towards this direction. Here, when it gets within that certain threshold, that dark blue trap is turned off and the other one is turned on. And so now there's even a stronger force pulling it there because it's like a spring force and the spring is extended, and then it goes in that direction. So my student from a few years ago, Shanshan Jiang, who's actually now working as an executive in a bank in Alabama, she just wanted to simulate this, and these are 2D simulations, and where you're seeing here, so the red object is actually, it's like a cell, it's got a very stiff membrane, and the forces that are exerted, this is Stokes flow, are forces due to this activated trap. So it gets close enough, that turns off, and the other one turns on. And you can see that that red dot is getting a little deformed because it's a membrane. Okay, so this is a simulation of a colloidal oscillator in Stokes flow. And it's going back and forth, and you could change its frequency by changing the viscosity of the fluid, by changing the stiffness of these traps, like these spring stiffnesses, and changing the threshold and so on. So you could tune this oscillator. So I have 2 minutes. I want to just show you something really curious. So then the question is, what happens now if you put 2 oscillators, 2 of these systems next to each other, coupled only through the fluid? So, I'm not going to tell you about the model, but what I should say is what I showed you there was Stokes flow, but computationally we wanted to check what happens if we added some inertia. Okay, so I'm going to spend 2 minutes on this, and then we'll basically be through. So now this movie might might get you very dizzy, so I have to tell you what you're seeing. This is a Stokes regime. You're not seeing the inactivated trap. So the blue things here are what the red particles are drawn to. When they get close enough, it's going to turn off, and its brother is going to turn on at a specific place here. Okay? So the question is, and I can't do this with a microphone, what's going to happen if you have 2 identical oscillators where they're at the moment placed exactly at the same position relative to its trap in Stokes flow? Or what happens if you perturb it a little bit? Are the 2 things just going to oscillate in synchrony like this? That's what I thought was going to happen with this calculation. Now, I should tell you that the physicists know the answer to that. So I'm going to show this to you and explain to you the craziness of Stokes flow yet again. So let me see. Let's see what happens. So here, I told Shanshan when she showed me this simulation that she had a bug in her code. Okay. Okay, and I promise it's going to get you dizzy. So what is happening here? So if you don't want to get dizzy, ignore the dark blue blobs. Just look at the red. Right? They're oscillating like this. They were in phase, and like almost immediately, they managed to get into antiphase. Look at that. Okay, so there was no bug in her code. And I'll tell you what happened. This is quite a stable equilibrium attractor antiphase here. And so what's going on? So let me start from the beginning again, because this is actually interesting. Well, everything I've said is interesting, but this is super interesting. So this is Stokes flow. It's going to happen that there's some numerical noise. This one here, this red, reaches its trap 1 time step before this one does. Now remember, there's no memory. So both of these red dots are going towards the blue dots. They're almost there. This one gets there first. This one gets there first. Immediately, this turns off and turns on here. That red guy, it's Stokes flow. He has no inertia. He does not remember, oh, I'm almost there, I'm going to continue going. As soon as this trap gets turned on in this direction, these are like spring forces. This red dot, there's such a big force pulling it here that the velocity, the fluid velocity that this big force induces does not allow the red dot to get to the blue one. Okay, so now that I said that, you can watch this one more time, maybe, and you can see what's happening. All right? The red guy back there got stuck, and then almost immediately it finds this happy equilibrium. So this was and this was realized in lab experiments at Cavendish Lab. So, here is a simulation too, where now the oscillators are not next to each other, but one's on top of the other, and Similarly, you can, if you just look at the red dots, you will see antiphase really quickly. So, and then, oh, this one you'll really like this. What if you have 3 in a row? So I have to do these movies in a better way because they get me very dizzy. But what you can see is that the neighbors become in antiphase and the ones at the end are in phase. So what I want to end with is this. When I told you that story walking through the simulation before, I said that one guy switched, but the other one didn't, but he didn't remember which way he was going. So instead of putting the simulation in a Stokes regime, I'm going to put in a Navier-Stokes solver. Of course, then we're using a whole other methodology to solve it, but I'm not going to tell you about that. We'll use an immerse boundary method. So here we have some small amount of inertia, and we still get opposite phase. Here we tune it and put a little bit more inertia so they know where they're going. And you see that the equilibrium is in phase oscillation. So this is an interesting example where this engineered physical system where Reynolds number or inertia. So here, if we put in a fluid of less viscosity in these lab experiments, it should be that you might not see the opposite phase attraction. So there's been a lot of analysis from these point oscillators, rotary oscillators, to try to say something about biological synchrony. And it's unclear to me what these systems that I've showed you really have to do with ciliary oscillation. But anyhow, this is a good place to stop. So think about these colloidal oscillators and the stability of in phase and opposite phase. And the moral of the story is Reynolds number sometimes really matters. Thanks.
Question?
When you, in the previous problem with the flagellant, when you say apply torque, is that still in the same model where the velocity of the particle is the same as the fluid velocity, or is this a different model?
Yeah, so what we do is we don't treat the point at which we're applying the torque as a material part of we don't update that with the fluid flow. We update the position of the helical surface, and then at the beginning of a time step, we say we're going to apply a torque at the centroid of its first section, and then we're going to go out a distance h normally and apply a counter torque.
Okay, so the torque is really a velocity that you...
It's in the forcing term, it's the curl of something. Yeah. So we're not updating where the counter torque is, because that counter torque will fly away. So we update, we satisfy the no slip condition on the helix, and then we say, okay, now torque on its nose and a little bit in front.
But the torque is an imposed velocity.
Yeah, so that's the thing that's we're imposed. But we're imposed, I don't know, we're imposing, a torque is this force. We're not imposing a velocity of rotation on the helix. We're imposing this. That gives rise to an angular velocity.
Because in the very first model, I had this question that if, is there some dissipation? I mean, can you, the system, the first talk, like the first,
The sperm flagellum.
That is a dissipative system?
Yeah, I'm not exactly sure what you mean, but we're keeping track of elastic energy the entire time. And so we don't have, if they're Hookean springs, there's no there's no dash pot in the Hookean springs.
Because they moved forever without any external force. It seemed.
Which
Last
does?
time, the things moved and bounced to the wall, and then but they never stopped.
We were driving it. I mean, we were in the sperm simulations of where we model things by the center line. There was an energy that was driven by a preferred curvature, which was a temporal traveling wave. So we never turned off that bend. We were driving it continuously. So here, we're only driving it at the nose. I don't know if that answers your question, but like in the viscoelastic mesh that I showed you, if we used, instead of Hookean springs, that always, that if you pull them out, they always want to go back in. If we used spring elements that had dash pots in them as well, like a Maxwell element, we could pull it out and then it would relax because we were putting in dissipation. I don't know if that answers your question, but yeah.
How do you do your simulations? What do you use?
Oh, okay. So what I did a couple of days ago, I described for the Stokes flow simulations, I'm using what we call the method of regularized Stokes slits, where it's a relation. So what that is based upon is, for the Stokes equations, if you have a single Dirac delta point force, you have an analytic solution, right? So instead of having a delta function point force, we regularize it a little bit, which regularizes the problem, and we have an analytic solution. And then we represent forces due to all these elastic elements and so on. We apply them to a fluid, and we have an analytic solution. It's not without its complications, because that's an n-body problem. Because you discretize, like in the helix, I have a bunch of 300 sections, 8 points on a section. You do the multiplication. I'm applying m forces. I'm computing the velocities due to those forces. That's an n-body problem. We use fast methods, and I didn't show you that here. Now, for when I told you that I added Reynolds number, I added inertia, that was a Navier-Stokes solver, an immersed boundary finite difference formulation. So yeah, it's always complicated to give talks like this. I'm not apologizing, but I've happily swept all of the numerical intricacies under the rug here. Yeah, so if that's your thing, I'm sorry. Some people, the movies are cool, but the numerics is pretty cool too.
When you have two-plug area, how do you work with the collisions or?
Oh, okay. That's a good point. So, what should happen if you had the perfect time integrator? You should never get collision because of, just because of no slip. But you have finite time steps, so you could. In fact, we can do our time steps in all of these simulations are, because we're using explicit stepping, our time steps are really small. So we hardly ever get collisions. One can put in repulsive forces. You can check to see whether things are getting too close, and you put it, I showed you this little vignette of the bundling, and there we would have to put in repulsive forces.
So for the first part of your talk, does the shape of the helix that forms from these flexible elements, is it uniform along the length of the?
No. In fact,
you have a different angle along the helix, right?
Yeah, so we never did really detailed shape analysis of that, but here's a good art show Here's an example.
These are simulated, right?
These are the simulations. This is the rigid helix, and the thing that looks like a champagne glass is the center line during one rotation.
Sure.
And so the specified shape, it should just have an amplitude, or this helical radius should be increasing linearly up to a point and then constant, pretty much. This is the achieved shape under the highest torque. So what you see is this is the emergent shape, and this is what the helix center line would look like during one revolution. And it is kind of interesting that that you don't get a constant radius out here. You get a maximum radius in a couple of places along here. So we didn't do any detailed analysis.
And the tail is at the center of
Yeah.
the head, or the virtual head. It's not.
Yeah. So this is a cylinder, and we put a torque at the centroid of this first section. And the head's not, the head's not really there. And it's hard to even, I'm happy this paper appeared, and I'm happy nobody ever said, well, how does the distance at which you put the counter routlet depend upon the size of the head? Because in fact, it's not like, okay, if there's a bigger head, put it further away. Because if you put that counter routlet further away, in fact, there's less drag on the system and not more. But it's a model, for all the students, when you have a model or so on, as long as you're honest about what it is, then I think it's fine. You don't argue that, yes, this is my model and it accurately represents that system because it might not. But as long as you're honest with what you're modeling.
So, and what I asked you earlier is that, like some flagellum, like for sperm, they're thicker at the base.
Yeah.
So you can
Yeah.
include that by changing the stiffness of your springs.
That's right.
As a function, and then.
Yeah. Who knows?
One thing would be interesting to see is what is the most efficient shape, because maybe it's too thick at the base and then too thin at the bottom.
Mm-hmm. So we could change the stiffnesses. We could also change, because we're actually representing the surface. We could change the surface. It could actually be thinner.
Yes.
Right.
Great.
Give me the microphone. I just want to remind, what was the number, the K, that were in the simulation program?
Oh, the K was just some non-dimensional stiffness of the spring.
Okay, stiffness of the spring.
Yeah. Yeah, so it's not, K equal 1200, it's not in any real unit. It was all non-dimensional. It was in program units.
Final question. In the experiment, in the cube, what's the border condition of the fluid in the wall?
In the simulation, zero velocity, no slip. So we put a bunch of points, discretized there, and asked and satisfied U equals 0 on those points. Right.
But you expect that the behavior is going to be different if you change the border condition?
Yeah, I mean, it could be. It could be. It's just it gets computationally very expensive, because the way that this works, just to let's see. So here's where I kind of wrote this out. We're adding a wall. So what happens? We applied a torque. We have some elastic forces here. Those forces, that when we get those, they're going to induce some non-zero velocities all over the tube wall, right? So now we say, okay, we have to then find out what forces are being applied at all these points on the tube wall to cancel out, to give negative those velocities. And we solve that linear system for those forces. And now we have forces on the structure and forces all along the tube wall, and those forces then give rise to the velocity to update the system. But when I say it's expensive, here, this is the dot. It's expensive, and that's where judicious programming skills come in. So in regularized Stokeslet, if you're just applying forces and you want to compute velocities, we have had undergraduates work on doing fabulous things, and they could program this in 3 days, not even. MATLAB code like this. Once you have to solve linear systems, now you could do MATLAB code, right? You could do A/B, but this will kill you. You can't do it on MATLAB because you have so many, if you're discretizing a very big tube. So.
Questions?
Thank you. Thank you. Thank you. This has been a couple of really fun days. Thanks for coming.